So last but definitely not least, we have Gina Saizan with the Louisiana Oil Spill Coordinator's Office. Gina, there you go. All right, so Gina's fun fact is that she has over 2,000 hours volunteering as an aquarist at the Aquarium of the Americas in New Orleans.
That's true. Forgot I gave you that one.
Thank you.
So Emily called a few weeks ago, 2.5 weeks ago, and said, is there anything missing from this agenda? And there was an open spot, and I said, yeah. I gave her quickly several species to maybe think about, and I started looking for speakers that could maybe speak to those topics and started putting feelers out. Nobody bit. And so then she said, well, I really want you to do it. And I'm like, oh, the lineup is pretty tough and I can't compete with that. So none of this is my research. It wouldn't have happened at all, but for Matthew Morschbacher with our office. So I want to thank him for pulling all the literature together. My normal activities are oil spill response, NERDA, and lots of coordination of trustees and agencies during a spill. So what I do have is experience out there. I think James Harris said it earlier, the locals have the majority of the knowledge, right? They know about those things. Well, I consider myself kind of a local of Marsh, South Louisiana, and even inland. I'm getting more and more familiar with inland Louisiana, just being out there, knowing what I'm looking at and seeing what things are happening during an oil spill. So that's where my knowledge base comes from, experience. Underdog, I was happy to see Kayla had that in her presentation earlier. That's the era that I came from, around the 70s, 80s. So his slogan was, have no fear, Underdog is here, right? So bioindicators, I'm not gonna read these slides because you guys can read it, they'll be online, so it shouldn't, you could go back and look at it. But we've talked about bioindicators multiple times today. They tell us about the environment, what could be happening out there, their attributes, their distributions, known biology, ability to provide that early alert, things like that. So Sentinel species, along those same lines, reliable ones are common, easily handled, and consistently measured responses. Examples, everybody knows about the canary in the coal mine. That could be a little hero, he's gonna fly into that coal mine, save human lives, but sacrifice the bird itself. It doesn't fly back out, it's too dangerous to go in. So the underdogs of Louisiana's bays and estuaries, among all the things we talked about today, I think mysid shrimp, grass shrimp, mussels, and marsh minnows are some of those things that are also overlooked, not typically studied. So first few slides are just going to be the biology of each one, and then maybe some of the literature that exists on these things. So mysids, my background, kind of first out of college, I always wanted to be the next Jacques Cousteau, right? I wanted to go and scuba dive and all those things. Well, I ended up culturing fish and shrimp to go into tox tests. And it kind of broke my heart because I was rearing these beautiful little fish to go be killed in toxicity tests. So it was kind of a dilemma for me. But I would count mysids my whole life. For about 3 years, I did that. And you can, when you feed them, you can, we fed them brine shrimp knobfly, and so you could watch the brine shrimp, the colored brine shrimp, red brine shrimp, going through their clear little bodies, and I just thought it was so fascinating, looking at them under dissecting microscopes. They're small crustaceans, they're abundant, usually in less than 6 feet of water. They're omnivores, they graze on all the epiphytes that are on the marsh plants and on the oyster beds and things like that. They're easily cultured, but somewhat sensitive. And they're used in EPA toxicity tests for effluent waters all the time. Drilling mud tests, things like that. So we have lots of good LC50 data, dose response curves on effluents, and most of those have PAHs in them. So we have some good information, but no Louisiana specific field studies on mysids that I'm aware of. Some of the effects, say that because they're clear body organisms, if they're exposed to PAHs, especially chronically, the PAHs are absorbed into their tissues, and the ultraviolet light in the shallow water, the penetration of the UV, actually causes more harm than just the PAHs in and of itself. Another factor is weathered oil. Here, this statement, weathered oil will substantially increase toxicity. And that's something that I found very interesting, because deep water was weathered as it came in. A lot of the spills that we see, people say, well, that's just sheen, it's weathered oil, it's not a big deal. Well, effects of weathering. So certain PAHs are phototoxic, severe tissue damage. This is just some of the literature that supports that mysids are affected by PAHs. Grass shrimp are the same way. Clear-bodied, small crustaceans, a little bit larger, they actually feed on the mysid shrimp. They're detritivores, so secondary consumers. They break down that organic material, just like the snails do. They start breaking down the plants and the epiphytes, and they're attracted and live on those plant stems and oyster reefs. They're a key link in the food chain. That's a gravid female with her eggs, and they're a huge support of fishery stocks. Instrumental in that transport of energy. Again, we have lots of literature out there, because grass shrimp have been used in tox tests, and really good information about the narcotic effects on shrimp from PAHs. So naphthalines are one of the components that, when it's in the water and it's fresh, those it causes almost like a drunken effect, and so they're disoriented swimming and motions, and you can imagine if they're not able to jump away quickly from something, they're gonna get eaten faster, potentially. Lots of detrimental effects on hatching larvae. So the juvenile stages of these invertebrates are even more susceptible than sometimes the adults. Chronic exposure, again, that's a reoccurring theme. Chronic exposure is usually more of a problem than acute. So over time, when these oil spills sit there over time. RHIB mussels. You see these in the marsh. Folks tend to walk over them, especially when there's high water. You don't see them readily, but they're there. Intertidal filter feeders, so they clean up the edge, right? They're there all the time, and they're basal threads. If you ever tried to pull one out of the marsh, they're intertwined with the Spartina grass, and they're extremely hard to get out, and they support that first wall structure of the marsh, so that edge, and they're there to provide that support and help the plants stay in place, keep the sediments there, and provide that that structural integrity. They deposit fecal matter on the surrounding sediments, stimulates the grass growth. They increase marsh net productivity and stability, as I said. Very few studies in the Mississippi deltaic region. So we have lots of studies on other ribbed mussels in the Atlantic region, but not a whole lot of literature here. NOAA's Mussel Watch program has a lot of good information that we could tap into and use for a literature base in oil spills. This is what they look like at the base of the plant. This is actually in an oiled area. They're just embedded in there. Most people walk over them. They don't know they're there. So this is RHIB mussels in toxicity tests. So some of the literature, or not toxicity tests, but in research. And some of the literature tells how important they are. Estuarine filtration, soil strengthening. So changes, if you lose the bivalves, you may start losing that structure, that edge, maybe even some of the plants. Oil does cause, in some cases, decreased growth rates, lower feeding rates, things like that. Marsh minnows. So two types here, the sheep's head minnow and Gulf killifish, but this is the sheep's head minnow. Gulf killifish, male and female. They're small. They live in our fresh brackish saline marshes. They're here all the time. They're omnivores, so they live off the epiphytes on the plants and the oyster beds, on the seagrass beds. They eat the small crustaceans, so the grass shrimp and the mysid shrimp. They spawn in spring and fall. The eggs stick to everything, the plant stems themselves, the oyster beds. So if you can imagine an oil spill occurring, the minnows laying their eggs on the plant stems, and then the oil coming in and affecting those embryos developing. So they're a very important prey for fish and birds, and I think the last talk really highlighted that. Maybe not the only thing, but an important part of it. They're a popular live bait. They have very small scale movement, so they don't go very far. There's not a huge home range there. Their sister species is studied pretty extensively. There's a lot of literature on other species, not a whole lot on this species in Louisiana, these two species in Louisiana. So this statement could become the marine science equivalent of the white mouse used in medical research. So lots of ideas here. So PAHs, you know, cause a lot of the same effects in the other things that we saw, maybe even more. So I think I've seen literature that supports that there's genomic effects that may be hard-coded into their DNA. And so the changes that are occurring through PAH exposure may be transferable in their DNA. So it's just something to think about. Whitehead and Fernando Galvez had lots of good data on gill histology. So CYP1A causing lots of changes in the gill structure, cardiovascular defects, delayed hatching, reduced hatching, all these things. So this is where I come in. I'm out there all the time. I'm seeing oil spills, and I know I'm on the periphery of the literature and know some of what's occurring in the research labs. But when we see this is oil, fresh product at the shoreline, coming out of a well, you know, hitting the shoreline. As the tide rises, this oil that's here is going to be pushed back into the marsh. And this is where everything is. All the things we talked about just now live at this edge and throughout the marsh. So this is a spill similar to that first picture where you had fresh oil. This is a little bit further in time, maybe about 2-3 weeks away. And we typically can still see this oil band here. So if you were to go up and touch that with a gloved hand, still a little sticky tacky, maybe not, you know, still washing off in the surf. So not free product, but still sticky tacky and transferable to organisms. And then right above that brown line, you can actually see the vegetation's chlorotic band. So it's being affected by that oil still. Maybe a few months later or up to a year later, sometimes that oil will actually cause this, which is dieback. I really enjoyed Scott's presentation where he said he planted, so that gives us some good ideas about what we might be able to do in areas like this. But when you get really close, and you know, this is a monitoring pool here where we've put a quadrat out. And this is what you see when you get close to it. That's actually footprints, which is not a good idea to get out of the boat and trample oil into the marsh or into the rootstock. We learned that from the work that Scott did in that cleanup area, the treated areas. that it's better to stay off that marsh. But to monitor it, to collect data, to help support NRDA and other things, we actually need to collect this. So this is, you know, information that we record and make note of if it's still out there months or years later. When you have that level of oiling in the sediments, you're going to typically see the die-off of the mussels, or even in the earlier days of the spill, the raccoons and the predators will come in, and because the mussels have been closed for some time, not able to feed, their basal threads are not as intact, the soil structure's not there to hold them, maybe even the plant rootstock is dying off, and they can easily pull out those mussels and open up and eat what's left. Again, a secondary contamination issue potentially. And this is what they look like once you collect them. That's how you can tell they've been broken up by a predator, probably a raccoon or something, a mink or a otter, sorry. And this is all the literature that we have access to, and Matt looked at to put some of this together. And it's, I mean, look at it, it goes on for days, but not a whole lot in Louisiana. And so underdogs are heroes. Can they be our heroes? You know, after one of the talks, I think it's nice to hear everyone's perspective, because, you know, I'm panicked when I look at some of this stuff, but it's a nice perspective to know, and it's reasonable to understand that Louisiana is resilient. We've had oil and gas production for over 100 years, and we still have, you know, beautiful marshes out there that are supporting a wide ecosystem of, you know, fish, birds, invertebrates. So it gives me a little bit of a calming effect to hear some of everybody's perspectives. So similar species are well documented in other areas, not as much here in Louisiana. Some species are well documented for other pollutants besides PAHs and oil. But, you know, Louisiana specific research, I think, I'm a scientist at heart, so I always want to know more, what's happening out there, and you know, how what I do on a daily basis affects these animals. And then the environmental impacts. Linda mentioned the hurricanes. We have systems that come through all the time. We have oil out there. We have extreme temperatures. Anybody that's been in the marsh in the summertime, a wide range of salinities. We got the freshwater diversions happening, all that. So what are those other environmental factors doing to oil exposed animals? You know, a cascading effect. And then there's always spills of opportunity. There's, Louisiana has between 1,500 to 2,000 notifications of oil releases each year. LOSCO is on scene for about 200 of those. Out of the 200, there's a very small number that actually move forward to a natural resource damage assessment. So there's always oil, you know, in Louisiana that's being released to the environment that we might be able to capitalize on and study. So just think about that, and you know, continue to research, folks. Thank you for that. I do have one anecdotal story, real quick. At the toxicity lab where I used to work, you know, again, it was against my better judgment to kill these animals that I raised, right? They were my babies. I didn't want to see them die. Occasionally, we had to put them in control type tests to make sure the animals were healthy. We had a healthy stock of them. And so we didn't have to kill those and weigh them out for the EPA tests. So I would take them after the test was over and they survived, and they were exposed to PAHs, but I would put them in tanks and rear them up. I didn't want to kill them. So I would watch how they were affected, and this is not a scientific study. I did it on my own time, took care of the tanks and fed them on my own time. But they actually grew up, and I saw a lot of the effects that we see with the Deepwater Horizon research that was done. You know, you had the fathead minnows and saprinodons that had the curved spines, and in the wild, I'm not sure they always had erratic swimming behaviors, and sometimes would have to be encouraged to eat. And so I think, you know, I was aware of all these things long before I became an oil spill biologist. But I believe in them, and I appreciate everybody's interest in them, and I appreciate Sea Grant for putting these things on, because I think James was right earlier. This is how we get the message out. The stuff that I'm seeing in the field, the stuff you're doing in the lab, you know, getting it, communicating, getting it out there, and so everybody's aware. Thank you.